Welcome to Web Microbiology, a podcast for Bio 204 at Allegheny College of Maryland. I'm Professor Kelly. Let's get started. Episode 27, Mutations. Welcome to the last episode for Chapter 6. In this episode, we're going to discuss mutations. Mutation is any permanent change to the nucleotide sequence in a genome. Mutations can involve loss or addition of base pairs, or a change to a single or multiple nucleotides. They can be spontaneous, meaning a random change due to replication errors, or they can be induced, meaning they're caused by exposure to a mutagen, a chemical or a physical substance that interacts with DNA molecules and changes them. When biologists talk about mutations and phenotypes, they use the term wild type to describe the phenotype most often seen in nature. Any change or alteration can be considered a mutant. In this episode, we're going to focus mostly on the molecular changes to DNA, what causes them, what specific changes are called, and what can result from mutation. Mutagens can be any physical or chemical substance that causes a change to the genome. Some of them you already know about. Ionizing radiation and UV light can cause DNA changes, which is one important reason why sunscreen is recommended and exposure to radiation equipment must be limited and monitored. Some drugs are designed to mimic nucleotides and when inserted into DNA cause mutation. Many chemicals that cause mutations are also carcinogens, or cancer-causing agents. The Ames test is one way to determine if a chemical or drug under development may be a mutagen. It uses special bacterial mutants that can't make the amino acid histidine. It plates them on special media that doesn't have this amino acid, and the chemical in question is added and the plates are incubated. These bacteria should not be able to grow without histidine in their environment unless they mutate. So if growth occurs above the normal background level, the chemical in question could be inducing mutation, which means it might be a mutagen. The Ames test is an example of microbiology used for industrial application. Let's get into our discussion about mutations and what can actually happen to the genome. A point mutation is a change in a single nucleotide base. An insertion is the addition of bases, from one to many, in a place they weren't present before, and a deletion is the loss of bases, from one to many, from the genome. It shouldn't be surprising that smaller changes are usually less harmful than larger ones, but let's consider some of the outcomes of these changes. So, a point mutation occurs, and it happens to be in the wobble position of a codon. That's the last of the three bases. Even though the base is different, the amino acid is the same, so there's no change to the protein it produces. We call this a silent mutation. You can see it in the genome, but it doesn't make any noise, so to speak, so there's no evidence of it in the phenotype. If a point mutation does change the amino acid, that's a missense mutation. This could be a big deal, or it might not be a deal at all. It depends on how it affects the function of the protein, and remember, protein function depends on overall shape. A missense mutation could result in a fully functional protein, a partially functional protein, or a non-functional protein. It really depends where the change occurs. Now let's look at another point mutation that changes an amino acid codon into a stop codon. That could be a big deal, especially if it happens near the beginning or even in the middle of a gene. It means only part of the protein will be produced because there's a premature stop. We call this a nonsense mutation because the protein can't be formed in its entirety, so the code is nonsense. Nonsense mutations are occasionally only minor problems, if and only if they happen near the very end of the protein. That's the only way that the protein might still be functional with a few amino acids missing at the end. Now let's consider a different scenario where two nucleotide bases are added to the middle of a gene. Remember that codons are 3 nucleotide bases, so when you add or subtract any number of bases that is not a multiple of 3, you're going to change the reading frame. This is called a frameshift mutation, and just like with nonsense mutations, it's usually a big deal. Every amino acid beginning at the frameshift will most likely be changed. Sometimes frameshift mutations can also cause a premature stop to appear because of the change. A frameshift mutation is like deleting one letter from a sentence. Every word after that point becomes total nonsense. Take a moment and try this for yourself. Write down the simple sentence, "The cat ate the rat." If you remove the very first T, you're left with something that totally doesn't make sense. Heck at her at. Frameshift mutation can be devastating to a protein. So how do we avoid mutations? I mentioned in a previous episode that polymerases are really good proofreaders, and they fix errors during replication processes. Let's talk about some other ways DNA mutations are corrected. When UV light damages DNA, it does so by creating unusual bonds between pyrimidine bases. An enzyme called photolyase detects these dimers and corrects them, but it can only do so much. Badly damaged cells are beyond repair and will die. This is why pigmentation of the skin changes in response to UV exposure, or more simply put, a suntan is a natural protection against DNA damage from sunlight. Another repair that happens is called excision repair. Special enzymes can find mismatched bases and cut them out of the DNA strand. DNA polymerase I comes behind and fills in the gap with the correct nucleotide, and then ligase seals the strand. When the number of mutations exceeds the ability of the enzymes to fix them, however, permanent changes occur. So let's review. A point mutation is a single nucleotide base change, while an insertion or deletion either adds or removes bases from the code. A silent mutation changes the DNA, but not the amino acid sequence, so there's no change to the phenotype. A missense mutation changes one amino acid, and that has varying effects on the functionality of the protein. A nonsense mutation introduces an early stop codon, and a frameshift shifts the reading frame. Some of these can be relatively minor, but others can be deadly for the cell. Thanks for sticking with me through this complex chapter. I'll see you in the next episode where we start talking about viruses in Chapter 7.